Several of these animals experience conditions that would kill any other creature within minutes. What makes them extraordinary is not that they survive, it is how completely they shut down, and how completely they return. Wood frogs, Rana sylvatica, survive being completely frozen solid. When winter temperatures drop, ice crystals form inside the frog's body, in the tissue, between organs, in the blood. The heart stops. Brain activity ceases. Up to 70% of the body's water turns to ice. The frog remains in this state for weeks or months. When temperatures rise, the process reverses, the heart restarts, blood begins to flow, and the frog begins moving within hours. The mechanism involves glucose. As temperatures drop, the liver releases massive quantities of glucose into the blood. This cryoprotective glucose raises the concentration of the cell fluid, lowering its freezing point and preventing the ice crystals from forming inside the cells themselves. The ice forms between cells, compressing them but not rupturing them. Researchers studying wood frog cryoprotection are developing the same mechanism for human organ preservation. Organs stored with cryoprotective glucose last longer at lower temperatures without cell damage. Greenland sharks, Somniosus microcephalus, live for a minimum of 272 years, and the oldest confirmed individual, by radiocarbon dating of the island's nucleus, which forms at birth and does not regenerate, was estimated at 392 years, making it the oldest known vertebrate on Earth. At birth, a Greenland shark that will live to 392 years old would have been born in 1634, before Isaac Newton's birth. The sharks reach sexual maturity at approximately 150 years. They move slowly, typically less than 1 km per hour, and live at extreme depths and temperatures, below -1 degree Celsius in some cases, that suppress metabolic rate. Their longevity is not a special adaptation so much as the consequence of living in conditions where almost nothing degrades, cold, dark, and chemically stable. Turritopsis dohrnii, the immortal jellyfish, can reverse its life cycle after reaching sexual maturity. When stressed, by injury, starvation, age, or environmental change, the adult medusa reverts to a polyp, its sexually immature larval form, from which it redevelops into a new adult. It can repeat this process indefinitely. The reversion involves transdifferentiation, cells changing from one specialized type to another, which occurs in almost no other multicellular animal. Some salamanders can do it in limited contexts. No individual specimen has been confirmed to have lived indefinitely in controlled conditions. The process is hard to observe in captivity without the stressors that trigger it, but theoretically there is no biological mechanism preventing indefinite cycling. Naked mole rats, Heterocephalus glaber, can survive 18 minutes of complete oxygen deprivation, long enough for any other mammal to suffer permanent brain damage, by switching their metabolism from glucose to fructose, a pathway mammals use only in the liver, not in the brain. Under oxygen deprivation, naked mole rat neurons continue functioning using fructose as their sole energy source while the heart slows to a near stop. When oxygen returns, normal metabolism resumes with no measurable neurological damage. They also do not get cancer. In 40 years of research, no spontaneous tumor has been confirmed in a naked mole rat. The mechanism involves a high molecular weight form of hyaluronan that suppresses uncontrolled cell proliferation, 5 times larger than the human equivalent and apparently unique to the species. They live for 30 years, 10 times longer than mice of equivalent body size. Turritopsis dohrnii aside, the immortality prize for invertebrates also belongs to Hydra, genus Hydra, a freshwater animal approximately 1 centimeter long that shows no measurable increase in mortality rate with age. Studies tracking thousands of individuals over 4 years found no increase in death rate, no decrease in reproduction rate, and no accumulation of cellular damage markers, the definition of biological non-aging. Hydra cells continuously replace themselves from a pool of stem cells that appear to divide indefinitely without accumulating DNA errors. A hydra that survives predation, disease, and environmental hazards shows no intrinsic tendency to die. In controlled conditions without external threats, individuals have been kept alive for decades with no observed aging. Olms, Proteus anguinus, cave-dwelling salamanders found in the Dinaric Alps of southern Europe, live for up to 100 years, do not reach sexual maturity until age 15, reproduce approximately once per decade, and can survive without food for up to 10 years. Their metabolic rate is so low that the physiological processes associated with aging occur in extreme slow motion. An olm that is 1 year old is equivalent in biological maturity to a human infant of a few weeks. They are blind, eyes are vestigial and covered by skin, lack pigmentation, and navigate entirely by electroreception and chemical sensing in total darkness. A population of olms discovered in 1986 in a cave in Slovenia was still present and apparently unchanged when researchers returned in 2016, the same individuals, in the same locations, 30 years later. Bdelloid rotifers, class Bdelloidea, reproduce exclusively by parthenogenesis. There are no males. They have survived for approximately 40 million years without sexual reproduction, which evolutionary theory predicts should be impossible. Asexual reproduction causes gradual genetic degradation through accumulated mutations. The answer, discovered in 2021, is that Bdelloid rotifers routinely incorporate foreign DNA into their own genomes, from bacteria, fungi, algae, and plants, using a mechanism called horizontal gene transfer that is normally limited to single-celled organisms. They can also survive complete desiccation, being dried out completely, radiation doses that shatter their DNA into fragments, and resuming normal function when rehydrated from a dried state years later. During desiccation, the shattered DNA is repaired using the incorporated foreign genetic material. Mantis shrimp, order Stomatopoda, survive impacts their own bodies should destroy. The smasher species delivers a punch accelerating at 10,400 g, equivalent to a bullet, at speeds of 23 meters per second. This creates a cavitation bubble, a vacuum collapse producing a shockwave at 4,500 Kelvin, that delivers a second impact even if the first misses. The mantis shrimp's own club absorbs these forces through a helix structure in the interior, the same geometry subsequently used by Boeing engineers for impact-resistant aerospace panels. Mantis shrimp clubs have been tested to withstand 50,000 strikes without fracturing. An equivalent human fist, proportionally, would need to be made of a material that does not yet exist. 8 animals. 8 systems for surviving what should not be survivable: frozen solid, oxygen deprived, completely dried out, living longer than recorded history. Tomorrow's animal has been hiding an ability nobody expected was possible. This channel posts a new animal fact video every day. Subscribe so you don't miss the next one.